Transcription is the first stage in gene expression in which RNA is synthesized. There are many types of RNA. Throughout this course, we will cover the following 5 major types of RNA. Ribosome RNA, or rRNA, is the most abundant RNA. Messenger RNA represent the most diverse RNA. They are subject to gene regulation. Some are very stable, while others are not. Transferred RNA is critical for translation. Small nuclear RNA is key to RNA splicing. MicroRNA plays an important regulatory role in the cell. Today, we will focus on 5 topics. The first topic is about transcription in prokaryotes. Second topic is about transcription in eukaryotes. Third topic, we will discuss processing of eukaryotic mRNA. For the fourth topic, we will discuss synthesis and processing of tRNA. And the last topic is about synthesis and processing of rRNA. DNA and RNA synthesis are similar. They all share general steps of initiation, elongation, and termination, with a new strand synthesized in the 5' to 3' direction. Both involve large initiation complexes. Both adhere to Watson-Crick base pairing rules, but they are different processes. RNA synthesis does not require a primer. RNA synthesis, only a portion of the genome is transcribed. In RNA synthesis, there is no proofreading. U replaces T as the complementary base pair for A in RNA. Ribonucleotides are used in RNA synthesis. During transcription, the sequence of ribonucleotides in an RNA molecule is complementary to the sequence of deoxyribonucleotides in one strand of DNA molecule, as shown here. The strand that is transcribed or copied into an RNA is called template strand. This is a template strand. The other strand, the non-template strand, is frequently referred to as coiling strand. It is called coiling strand because, with the exception of T for U change, it corresponds exactly to the sequence of the RNA primary transcript. In the case of a double-strand DNA containing many genes, the template for each gene might not be the same. For example, there are 4 genes here on this DNA. 3 genes use the bottom strand as the template strand, while gene C uses the top strand as the template strand. RNA polymerase move in 3' to 5' direction along the DNA template strand. RNA is synthesized in the 5' to 3' direction. This picture shows you the start and end of the template strand for a particular gene. RNA synthesis starts here and ends here. At the beginning, RNA is very short. Bend is very narrow. When it gets close to the end, RNA gets longer and the bend is getting wider. DNA-dependent RNA polymerase, or simply RNA polymerase, is the enzyme that synthesizes RNA based on the DNA template. The enzyme attached at the specific size on the template strand which is called the promoter. This is followed by initiation, elongation, termination. Transcription unit is defined as a region of DNA that includes signals for transcription initiation, elongation, and termination. The RNA product is called primary transcript. Conventionally, base sequence of a gene is given from coding strand in the direction of 5' to 3'. The first base, transcribed as RNA, is designated as +1 base of the transcribed region. To the left, upstream of 5' base are named as -1, -2, and -3, and so on. To the right, downstream of 3' bases are named as +2, +3, and so on. Now, let's go to part 1. Prokaryotes have only one RNA polymerase that synthesizes all types of RNA in the cell. Core enzyme have 4 subunits, including alpha and beta subunits, reminiscent of hemoglobin, but it has 2 different beta subunits. As a result, it has alpha-2, beta prime composition. Initiation of transcription requires sigma factor. Sigma factor recognizes a specific site in the promoter of a gene and brings RNA polymerase to it to start transcription. Promoter has 2 consensus sequences. One sequence is located 10 nucleotides upstream of the transcription start site. It is called pre-mRNA box. Second sequence is located 35 nucleotides upstream of the transcription start site. It is called -35 sequence. Core enzyme of RNA polymerase have alpha-2 beta prime composition. If we add sigma factor to it, we will have a hollow enzyme. Bacterial transcription occurs in 5 steps. In step 1, RNA polymerase hollow enzyme bind the DNA and find the promoter. In step 2, hollow enzyme make a dinucleotide with the first nucleotide being adenine. In step 3, after RNA chains reach 10 to 20 nucleotides long, RNA polymerase undergoes a conformational change and is then able to move away from the promoter and transcribe towards downstream. In step 4, successive residues are added to the 3' end of the nascent RNA molecule. In step 5, when RNA synthesis is complete, RNA chain and RNA polymerase are released from the template. Details of these 5 steps are provided on this slide. Now, let's take a closer look at a few key steps. For initiation, hollow enzyme, scanning through the chromosome to find the promoter region. Once it finds the promoter sequence, sigma factor will dissociate and RNA polymerase will start RNA synthesis. When RNA polymerase binds to a promoter, it causes 2 DNA strands to be separated. In this region, a short stretch of nucleotides are exposed on each DNA strand. This entire structure is called transcription bubble. When RNA polymerase moves forward, transcription bubble will move along. Opening up double strands called supercoil ahead of the transcription bubble that need to be unwound in order for the RNA polymerase to pass through. On the other side of the transcription bubble, 2 strands need to be rewound to bring the DNA back to double helix. Unwinding ahead of transcription bubble is carried out by DNA gyrase. Rewinding behind the transcription bubble is done by topoisomerase I. There are two types of transcription termination mechanisms. First mechanism is role-independent. The mRNA contains a sequence that can base pair with the cell to form a stem loop structure, 7 to 20 base pairs in length, that also rich in GC base pairs. Here is a stem loop structure. In the stem, the nucleotides form base pair. In the loop, they are not base paired. Stem loop structure also sometimes called hairpin structure. Second mechanism is role-dependent. Role is an ATP-dependent helicase. Role recognizes a specific sequence on RNA. It moves along RNA and unwinds the RNA-DNA hybrid duplex structure. Eventually, it displays RNA polymerase from the transcription bubble. There are two features in prokaryotic transcription. First, coupled transcription and translation. Translation and transcription occurs simultaneously in bacteria, while RNAs are still being transcribed from DNA. This picture shows you RNA in red still being made. Translation has already begun. This picture provides evidence to demonstrate the coupled transcription and translation in bacteria. Here is the transcription start. Here is transcription ends. At the beginning, transcript is short with fewer ribosomes bound to it. The band is very narrow. When it gets closer to the end, the transcript gets longer with more ribosomes bound to it. The band is wider. Second feature, transcription in bacteria is both monocistronic and polycistronic. This is the example of monocistronic. One gene on the DNA is transcribed into mRNA, and at the end, only one protein is made from that mRNA. This is the example of polycistronic. One gene from the DNA is transcribed into one messenger RNA, and this mRNA encodes several different polypeptides. And this is polycistronic. Now, let's switch to part 2. These are the requirements for transcription in eukaryotes. As you see, this is very similar to prokaryotes. Major difference is in eukaryotes, more proteins are needed to perform one particular function that is done by one protein in prokaryotes. For example, there's only one sigma factor in prokaryotes. In eukaryotes, there are many transcription factors in place of one sigma factor. Another example, in prokaryotes, one RNA polymerase synthesizes all types of RNA, but in eukaryotes, there are 3 different RNA polymerases. RNA polymerase 1 synthesizes rRNA in the nucleolus. RNA polymerase 2 synthesizes heterogeneous RNA, mRNA, small nuclear RNA. RNA polymerase 3 synthesizes tRNA, 5S RNA, and small nuclear RNA. In eukaryotes, all mRNA is monocistronic. Transcription in eukaryotes can be described in 3 steps. In step 1, transcription factors help RNA polymerase II recognize and bind to the promoter region. Promoter in eukaryotes has 2 consensus sequences. One is Tata box, found at 25 base pair upstream of the transcription start site. Second sequence is CA80 box, found at 70 base pair upstream of the transcription start site. As you may recall, in prokaryotes, they are much closer to the transcription start site. In step 2, RNA polymerase separate the 2 strands of DNA and create transcription bubble. RNA polymerase reads the template strand in 3' to 5' direction and synthesize RNA in the 5' to 3' direction. The RNA synthesized at this point is called a primary transcript. In step 3, RNA polymerase ends transcription when it reaches termination sequence. This picture shows you RNA is being transcribed from a gene on the DNA. Transcription results in a primary transcript that contains both exons and introns. Introns will be cut out later. This slide shows you assembly of the basal transcription complex around a promoter. Here is a gene region, and here is transcription start site. This is Tata box, this is RNA polymerase II, and this is transcription factor TF2D. The first step in transcription is binding of transcription factor TF2D to the Tata box, followed by 2B. These two together recruit RNA polymerase II, along with 2F. 2 more proteins are going to join them. One is 2E, another one 2H. Now, the basal transcription complex is ready to transcribe RNA. These slides show you the same initiation process in another way. These slides highlight the importance of TF2D in initiation of transcription. TF2D is a protein complex comprised of a Tata binding protein, or TBP, and several other proteins called TBP associated factors, or TAF. Some genes don't have a Tata box. In such cases, 2 alternative sites for transcription initiation are initiator sequence and the downstream promoter element, or DPE. Initiator sequence is located at the transcription start site. TAF1 and TAF2, 2 subunits of the TF2D, bind to the initiator sequence and direct RNA polymerase II binding to the promoter. DPE is about 25 base pair downstream of the transcription start site. Similar to initiator sequence, DPE sequences are also bound by the TAF subunit of the TF2D. Sometimes we need a high level of gene expression, such as those genes that control embryonic development. This is achieved by enhancer. It is a DNA sequence, single or multiple copied in either direction. Usually, it is located upstream of the gene, but it can be downstream. It can be far or can be close to the gene. Mechanisms that counteract enhancer also exist, that include silencer elements, a particular DNA sequence, and repressor proteins. Repressor proteins bind enhancer, suppress transcription of the target gene. This is integration with pharmacology. A group of antibiotics related to rifamycin block RNA polymerase. It has been used to treat tuberculosis. Actinomycin D binds DNA and blocks movement of RNA polymerase. It has been used as an anti-cancer drug. Alpha-amanitin, a toxin from certain mushrooms, blocks RNA polymerase II, leading to cell death. Now, let's go to part 3. In eukaryotes, primary transcripts need to be processed before being exported into cytoplasm as mature RNA. RNA processing includes the following modifications. First, methylated cap is added at the 5' end, as shown here. Second, poly-A tails is added at the 3' end. Number 3, introns are removed. And number 4, alternative splicing. Here, I need to introduce you two terms: primary transcript and heterogeneous nucleus RNA, or hnRNA. Primary transcript is the single-strand RNA product synthesized through transcription of DNA. Heterogeneous nuclear RNA is found in the eukaryotic nucleus associated with proteins to form higher-order structures. Not all primary transcripts are heterogeneous nuclear RNA, but those need to be processed are heterogeneous RNA. Here is detail of 5' capping. This is the 5' end of the newly synthesized RNA. Guanosine is added to the 5' end backwards. Methyl group is added at position 7 of the purine base. Methyl group is also added at 2' position on the ribo of the newly synthesized RNA. 5' capping improves translation and increase stability of the mRNA. RNA polymerase II does not terminate at distinct site. Instead, the pre-mRNA is cleaved 20 nucleotides downstream of this poly-A signal, and then about 250 A are added to the 3' end. The result is poly-A tail. Some genes contain introns. Introns must be removed before the final RNA exits from the nucleus. This process is called RNA splicing. Introns are removed from heterogeneous nuclear RNA by spliceosomes that are complex of small nuclear RNA and proteins. Here is a spliceosome. RNA will be spliced. Proteins associated with small nuclear RNA are also called small nuclear ribonuclear proteins, or SNRP. Splicing is accomplished in 2 steps. In step 1, 2' hydroxyl group of a specific adenosine in the intron branch point attacks the 5' splice site. This leads to formation of a lariat. In step 2, 3' hydroxyl group of the upstream exon form phosphodiester bond with the 5' end of the downstream exon. Lariat is released and degraded. As a result, the intron is removed. This is an example of extensive splicing. The dystrophin gene is very large on the DNA. Transcription leads to primary transcript with 2.5 million nucleotides. Splicing yields mRNA with only 14,000 nucleotides. Almost 95% of nucleotides are in intron and therefore removed. Here is the clinical correlation. Incorrect splicing accounts for 15% of genetic disease. Beta thalassemia is one of them. We have discussed this disease before. Today, we will only focus on the cause. Incorrect splicing causes non-deletion form of beta thalassemia. For some genes, there's more than one way to splice the primary transcript. Different way to splice an RNA leads to different RNA products. This is called alternative splicing. The advantage of alternative splicing is to generate a wide variety of proteins from a small number of genes. Here is an example. Fibronectin. Fibronectin can be spliced differently in fibroblasts and hepatocytes. The full-length mRNA is produced in fibroblasts. Shorter transcripts are made in hepatocytes and few other cell types. Now, let's move to part 4. tRNA is synthesized by RNA polymerase III. The pre-tRNA contains 5' leader sequence, an intron, and 3' action nucleotides. Post-transcriptional processing of tRNA includes removal of 5' leader sequence, introns, and 3' action nucleotides. Addition of CCA at 3' end, and base modifications. In some positions, G, A, C, U are modified. For example, uridine is converted to pseudouridine, ribothiamidine, and dihydrouridine in some positions. These slides recapitulate modification on tRNA. Removal of 5' leader sequence, introns, and 3' action nucleotides, addition of CCA at the 3' ends, and base modifications. Here are some unusual bases seen in tRNA. Most occurs on uridine, such as ribothiamidine, pseudouridine, and dihydrouridine. Post-transcriptional folding and base pairing give the characteristic clover-leafed shape of tRNA. tRNA has 4 main arms: D-arm, T-arm, acceptor arm, and anticodon arm. Some tRNA has an extra variable arm. Typically, each arm has one stem and one loop. For example, the D-arm has one stem and one loop. A stem is a base-paired segment of hairpin structure in RNA. A loop is a single-stranded region at the end of a hairpin in RNA. Amino acid is attached at the 3' end of tRNA. It is the anticodon on the anticodon loop that recognizes a codon from mRNA by base pairing. tRNA can form some unusual base pairing with mRNA. Here, base from tRNA are listed on the left and base from mRNA are on the right. For example, guanine from tRNA can pair with uracil from mRNA. Vice versa, uracil from tRNA can pair with guanine from mRNA. Inosine from tRNA can pair with either adenine, cytosine, or uracil from mRNA. Now, let's go to part 5. Eukaryotic 5S rRNA is synthesized in the nucleus by RNA polymerase III. The rest and much larger rRNA is synthesized in the nucleolus by RNA polymerase I. It is transcribed as a single piece 45S rRNA, then cleaved to yield 28S, 18S, and 5.8S rRNAs, each containing a number 8 in it that can help us remember them. In prokaryotes, the large and small ribosomal subunits are 50S and 30S respectively, which together make 70S ribosome. In eukaryotes, the large and small ribosomal subunits are 60S and 40S respectively, which together make 80S ribosome. Eukaryotic ribosomal RNAs are made from pre-ribosomal RNAs synthesized in different locations. RNA and proteins are assembled into 60S and 40S subunits in the nucleolus and then exported into the cytoplasm.